Assalamualaikum warahmatullahi wabarakatuh. Good evening, everyone. First, let me introduce myself. My name is Ayu Fitriani Rizkia Lestari, and I am a student of UNC'2023 University. Today, I want to talk to you about biotechnology. The purpose of this presentation is that students can understand the various types of biotechnology appropriately. My presentation is divided into three parts. First, I will talk about definition of biotechnology. Next, types of biotechnology. And finally, conclusion. Let me start with some general information of biotechnology. What is biotechnology? Biotechnology is a combination of two words, namely biology and technology. So we can interpret it as applied science and technology that utilize living things to produce goods and services that can be utilized by humans. Types of biotechnology. First, conventional to modern. Conventional biotechnology. Conventional biotechnology is biotechnology that utilizes organisms directly to produce product and service that are beneficial to humans through the fermentation process. First, cheese. In cheese making, lactic acid bacteria play a role in producing acid and flavor, helping card formation and producing lipolytic and proteolytic enzymes that are important in cheese ripening. The lactic acid bacteria used for cheese fermentation are of the genus Lactobacillus and Streptococcus. 2. Yogurt. Yogurt is a dairy product made by fermentation process using lactic acid bacteria, Lactobacillus bulgarius, and Streptococcus thermophilus. Next, butter. Butter is a dairy product made with the help of bacteria. Milk cream is fermented with the help of Streptococcus lactis and Lactobacillus bacteria. Next, tempe. Who does know tempe? Tempe is a food made from soybeans of several other ingredients that are processed through fermentation, use small microorganisms, rhizopus, or commonly known as a cedar or tepe, yes. Soy sauce. In making soy sauce, there will be a fermentation process that regulates the role of fungi and bacteria. Aspergillus soyae and Aspergillus oryzae are mushrooms that are widely used for soy sauce making. Tape. Tape. The process of making tape begins with fermentation that occur during the manufacture of sticky rice. Tape is not separate, and the role of microbes found in tape comes from mold. And bacteria such as Aspergillus oryzae, Saccharomyces, Rhizopus, Henzula, and Torolopis. Tauco. Tauco is a food ingredient from white soybeans formed by Aspergillus 20 bacteria with medium derived from the white soybeans. Agricultural biotechnology. Hydroponic. First, hydroponic, method of growing crops with water media. Three methods. First, culture of sand to water, and porous Jeffrey stone fragments. Next, aeroponic, the method of farming by sparing water, mist, and nutrients to the roots of plants. Modern biotechnology. Modern of biotechnology known of technique that involves genetic engineering so as to produce recombinant DNA and transgenic organisms that can be utilized to produce derived products such as EVA, hormones, antibiotics, and vaccines. Genetically, engineering, recombination, or genetic engineering is the process of change genes with the aim of obtaining new organisms that have the desired characteristic. Example of genetic engineering. First, DNA recombination, 2 cell fusion, 3 cord transfers. 1 DNA recombination. Every time of living thing has the same DNA. DNA from one species can be linked to DNA from another species in order to acquire new traits. This splicing process is known as DNA recombination. Example of DNA recombination: first, manufacture of insulin, to vaccine manufacturing. As you can see, guys, this is a picture of DNA insulin. Yeah, that makes the insulin. Next, cell fusion. Cell fusion or hybridoma technology is the process of melting or unitering two cells from the same of different tissues or species, so that a single cell containing genes from the two different cells is a product. Example of cell fusion: fusion of human cell with mice. B, fusion of tomato cells and potato cells. A human cell fusion with mice. Human lymphocytic cells are capable of producing antibodies, But if cultured and minded, the division process is very slow. Human cells are fused with mouse cancer cells with the aim of dividing quickly because mouse cells contain myeloma, which has the ability to divide rapidly. The hybridoma format will get antibodies, cell properties, humans, and are able to divide rapidly or a thread of mouse cancer cells. The combination of tomato cells with potatoes. Plant cell fusion is often called fusion between these plants. Plant cell walls composed of cellulose with must be destroyed by enzyme first. Then the protoplasm remains fused. For the example, tomato plants, which are new plants that produce tomatoes and tuberous potatoes. 3, core transfer or cloning. Nuclear transfer is the process of transferring the core of body into an egg cell without a nucleus, so that the egg cell will divide and become an embryo. Core transfer is also called core cloning or cloning. Example of cloning: a dolly sheep. Cell in nuclei taken from the mammary gland tissue of white-faced sheep. while the ovum is taken from a black-faced female sheep whose core has been damaged so that it becomes a nucleus ovum. Next, the cell nucleus of the white-faced sheep body is inserted into the ovum of the black-faced sheep and nurtures until it reaches the blastula stage. Then insert to the uterus of the black-faced sheep, and as a result, the sheep dolly was burned. And then yeah, that's a picture. And thank you. So now, so that's all about. In conclusion, let me sum up my main points. Well, that's it for me. I'm sorry. I'm sorry. In conclusion, let me sum up my main points. Biotechnology is the use of living things to produce goods or service that can be utilized by humans. Biotechnology consists of two types, namely traditional biotechnology or conventional and modern biotechnology. Traditional biotechnology includes the manufacture of tempeh, cheese, butter, etc. While modern biotechnology, example, manufacture of vaccine, insulin, and cloning. So yeah, well, that's it. Well, that it's all from me. Thank you very much. If you have question, you can comment below. Don't forget to like and subscribe. Dadah guys. Assalamualaikum warahmatullahi wabarakatuh.